Hello students, welcome to the new session. Today we will be discussing Science 2 Chapter 1. In this we are going to learn about heredity and evolution. You must be remembering what you have learned in 9th standard regarding heredity, that chapter 16, heredity and variation. So the first part of the lesson is just a beginning, just a recollection to what you have learned in 9th standard, and thus you are going to learn what are the changes or how do DNA helps in making protein synthesis and the characteristics are existing in us. So because of these proteins there are various characteristics. So that's what you will see the first part of the lesson. Later on you will learn how is evolution has happened. From billions of years ago the life was not there, but the life began to be existing, and thus we see different species which is around us. So we will be learning or discussing about all that in this chapter. So first and foremost, let's see about heredity. What is meant by heredity? Transfer of characteristics from parents to offspring, that is called as heredity. So here in your textbook, as the lesson begins, there are a few questions. Let's discuss what are they. Which component of the cellular nucleus of living organisms carry hereditary characteristics? We know it is in the nucleus, and that is chromosomes. So the answer is chromosomes. What do we call to the process of transfer of physical and mental characteristics from parents to progeny? The transfer of characteristics is called as, yes, heredity. Which are the components of the DNA molecule? The characteristics are being transferred because of chromosomes, or we say it as DNA. And so what are the components present in the DNA molecule? DNA full form is deoxyribonucleic acid. So there are molecules of deoxyribonucleic acid. There will be some phosphate molecules. There are also purines and pyrimidines. They are also called as nucleic acid. So what are purines? What are pyrimidines? If you forgot, you go through your chapter 16 of science 9th standard chapter, lesson. Okay. Now these are just an introduction to the lesson. Now going on to the lesson, the first part is about the history. So a few scientists' names I have written as it is given in your textbook also, and what are their discoveries, or in which way they gave us knowledge more about DNA. That is what is this. So some of the fill in the blanks are there regarding some questions related to this may also come. So you need to go through this history of heredity and hereditary changes, or the scientists who worked to know about this. Okay, so let's see the first one, that is in the year 1886. So who is that scientist? Gregor Johann Mendel. What did he do? He what is his contribution? Series of experiments he has done on pea plants to explain the inheritance of characteristics. So that also we have discussed in 9th standard. Well, the next scientist who came up in 1901, that is Hugo de Vries. His study topic was mutational theory. His discovery was, what is the reasons behind sudden changes? The reasons behind sudden changes was his study topic, or that was his discovery also. Then in 1902, Walton and Sutton, their study topic was chromosomes, and the discovery was paired chromosome in grasshopper cells. They found there are chromosomes, these chromosomes are paired, two chromosomes are actually paired, and then they discovered this paired chromosomes in grasshopper cells. That is in 1902. Then after that, in 1944, many scientists like Oswald Auray, Macklin McCarthy and Colin MacLeod, all of them studied again about DNA, and they discovered all living organisms have genetic material called as DNA, except viruses. That was their study. All living organisms have genetic material in the form of DNA. Then after that, in 1961, a scientist called Francois Jacob and Jack Monod, they studied about protein synthesis. How are these proteins synthesized in the cells? So they studied about it, and then their discovery was a model for process of protein synthesis with the help of DNA in bacterial cells. So this is the history of discovery of the hereditary or towards DNA. So we see many of the scientists studied regarding DNA. Once again, what is the full form of DNA? Deoxyribonucleic acid, that is DNA. It is nothing but acid, nucleic acids, or they are molecules. They are simple molecules. Now, do you remember the structure of DNA? I'll just put a clip here. You can see the structure of DNA. It is what is the name for that? Double helical structure. The structure of DNA is double helical structure. So there will be, it's like a twisted ladder, a ladder which is twisted. That is the shape of DNA. So it is having ribonucleic acids, and then there will be some phosphate molecules and purines and pyrimidines. So which are they? These nucleic acid molecules, purines, pyrimidines, adenine and thymine, cytosine and guanine. These are called as nucleic acid molecules. They are purines and pyrimidines. Adenine and thymine. Adenine will always join with thymine, and cytosine and guanine. So this continuously, these molecules are being arranged in the DNA, and part of the DNA is just a strand, a very long strand. A part of the DNA is called as gene, and so the gene is responsible for protein synthesis. When these proteins are being synthesized, we have that particular characteristic. For about your hair is black in color. It is because, or brown in color, it is because of that protein present in your body. So who is responsible for that protein synthesis? That gene is present, the gene helps in the protein synthesis. So this protein is synthesized, how it is being synthesized, that is what we are going to learn in the next part. So this is in 3 ways, that is transcription, translocation and translation. These are the 3 important ways in which the protein is being synthesized. So we know that in order to produce this protein, there has to be a gene, and this gene is present in the DNA. So part of the DNA. So there will be various genes in one chromosome itself, or one DNA itself. So out of that, only a part is responsible for making a particular protein, and if that protein is there, you have that characteristic. For example, some people are very tall or short. Tallness and dwarfness is due to a protein. So also the color of your eyes is because of a protein. So who is responsible for making this protein? The DNA is responsible for making the protein. So how is this protein produced? That is what you learn in these 3 processes, which are the transcription, translation and translocation. We know the process of the RNA synthesis is called as transcription. The process of RNA synthesis is called as transcription. From the DNA, the RNA is being made, and that is called as transcription. As you know that DNA has got 2 strands. like this DNA is. So one of the strand is responsible for making RNA. So here there will be there are either purines or pyrimidines will be there. So this nucleic acid, only one side will get will keep on making the complementary molecules, and thus that is called as the RNA. They are actually messenger RNA. Now see this protein synthesis because of DNA through RNA, this is called as central dogma. What is central dogma? Proteins are synthesized by DNA through RNA. This is called central dogma. What is central dogma? Proteins are synthesized by DNA through RNA. This process, this is called as central dogma. Now what is transcription? The process of RNA synthesis is called as transcription. Well, now you will see another important part of this transcription is triplet codon. Now what is a triplet codon? The code for each amino acid consists of 3 nucleotides. It is called as triplet codon. What is a triplet codon? The code for each amino acid consists of 3 nucleotides. This is called as a triplet codon. So you must have already come to know the process of making RNA from or by DNA is called as transcription. Now the next process is translation. What is this translation? Here each mRNA is as per the message of mRNA, amino acids are supplied by tRNA. For this purpose, tRNA has an anticodon having complementary sequence to the codon on mRNA. This is called as translation. So now mRNA is made, messenger RNA is made, transcription is done. What has to be now? We know that protein has to be made. For that, making proteins, there is a tRNA. So this tRNA will keep on making anticodons. What code they have got from this, against that another code is being made, which is called as anticodon. This is called as translation. So the tRNA makes an anticodon. This is called as translation. tRNA has anticodon having complementary sequence to the codon on mRNA. This is called as translation. Now let's see what is translocation. Now we know that the protein has to be made. So already the message has come. So only now molecule has to be arranged. So this process is taking place in the ribosome. So the ribosome keeps on moving through this messenger RNA and then keep on collecting the required molecules, and thus it will make a protein. So this is called as translocation. During this process, the ribosome keeps on moving from one end of the mRNA to other end by the distance of one triplet codon. This is called as translocation. What is translocation? The ribosome keeps on moving from one end of mRNA to other end by the distance of one triplet codon. This is called as translocation. Remember, messenger RNA is come from DNA, and now that is again translation has taken place, or anticodon is already made. Now what will happen? A ribosome will keep on sweeping over this messenger RNA, and thus it keeps on collecting molecules and makes proteins. This is called as translocation. You will be able to understand it with the video which I am playing towards it. At the end of this part is actually mutation. So due to certain sudden changes, there can be a change happen to this nucleotide, that is called as mutation. So sometimes any nucleotide of the gene changes its position, that causes a minor change which is called as mutation. So we have seen there will be adenine, guanine, thymine, cytosine, like this. These are the nucleotides. Any of this nucleotide changes its position, then there will be a slight change in the minor change can happen, and thus this is called as mutation. So due to mutation, there will be a new characteristics will take place. So by the end of this lesson, you understood what is heredity, which are the scientists who contributed study towards their work towards DNA and protein synthesis. Then we learned how is this protein synthesis taking place through translation, transcription, translation and translocation. And towards the end we learned what is mutation. Mutation is nothing but there will be some change in the positions of the nucleotides, keeps on changing due to sudden changes, maybe some kind of environmental factors. So this is called as mutation, and once mutation takes place, there will be change in the characteristic also in that particular organism. So this is what is regarding heredity of chapter 1 of science 2.
Here is a cell, the basic unit of all living tissue. In most human cells, there is a structure called the nucleus. The nucleus contains the genome. In humans, the genome is split between 23 pairs of chromosomes. Each chromosome contains a long strand of DNA, tightly packaged around proteins called histones. Within the DNA are sections called genes. These genes contain the instructions for making proteins. When a gene is switched on, an enzyme called RNA polymerase attaches to the start of the gene. It moves along the DNA, making a strand of messenger RNA out of free bases in the nucleus. The DNA code determines the order in which the free bases are added to the messenger RNA. This process is called transcription. Before the messenger RNA can be used as a template for the production of proteins, it needs to be processed. This involves removing and adding sections of RNA. The messenger RNA then moves out of the nucleus into the cytoplasm. Protein factories in the cytoplasm, called ribosomes, bind to the messenger RNA. The ribosome reads the code in the messenger RNA to produce a chain made up of amino acids. There are 20 different types of amino acid. Transfer RNA molecules carry the amino acids to the ribosome. The messenger RNA is read 3 bases at a time. As each triplet is read, a transfer RNA delivers the corresponding amino acid. This is added to a growing chain of amino acids. Once the last amino acid has been added, the chain folds into a complex 3D shape to form the protein.
Let's see the next part regarding evolution in the next session. Thank you for watching.